Hello, I'm Dr. Han, your instructor. I'm going to talk about Chapter 3, the cell. In this section, I'm going to focus on structure of the nucleus and some other organelles. There are two types of cells. One type is called a prokaryotic cell. The other one is a eukaryotic cell. The difference between prokaryotic and a eukaryotic cell is that eukaryotic cell has a membrane-bonded nucleus. Every eukaryotic cell, including plant and animal cells, all have this structure called a nucleus. Nucleus is surrounded by a membrane called the nuclear envelope. A double membrane wraps around the nucleus. This is called the nuclear envelope. On the surface of nuclear envelope, there are nuclear pores, which will allow certain molecules, such as RNA, to pass through. Within the nucleus, we have chromosomes, which contains DNA and the protein. In the center of the nucleus, we have a dark stained region called the nucleolus. Nucleolus is the place where the ribosome is being synthesized. Ribosome plays an important role in protein synthesis. In order for the DNA to fit inside the nucleus, the DNA is wrapped around the protein to form fibers called chromatin. The chromatin fiber twists and folds to form chromosome during the cell division. Chromatin and the chromosome are the same structure containing protein and the DNA. So chromatin will be invisible during the cell resting stage. However, during the cell division stage, the chromatin will become visible. At that stage of cell division, the chromatin will be called a chromosome. Humans have 46 chromosomes. Within the nucleus, there's a dark stained centered region called the nucleolus. That's where ribosome is synthesized, and the ribosome is an important component for protein synthesis. The reason we have a DNA because it's a control center. DNA is a template containing the genetic code. DNA can be transcribed into RNA, then RNA will be translated into protein. DNA is located within the nucleus, but RNA, once it's synthesized in the nucleus, it will be passed into the cytoplasm through the nuclear pore on the nuclear envelope. Then RNA will be translated into protein within the cytoplasm. This is the process of protein production. It is also the foundation for molecular biology. Other organelles involved in protein production include ribosome, which we mentioned earlier, and also the other one is endoplasmic reticulum. The ribosome will be located on some portion of the endoplasmic reticulum to make it rough. Then protein will be synthesized on the surface of it. Once the protein is being synthesized, it will be packed and labeled in an organelle called Golgi apparatus. In summary, the steps in protein production is DNA is the template which is located in the nucleus. He will be copied into a single-strand RNA through a process called transcription. Then RNA is going to leave the nucleus through the nuclear pore to the endoplasmic reticulum. On the endoplasmic reticulum, there are ribosomes on there. So the RNA is synthesized into protein on the ribosome. Then protein will be packaged into Golgi apparatus. Within the Golgi apparatus, he will be further labeled. Therefore, there are two terminologies you need to pay attention. One is the transcription in the nucleus. This results in the production of RNA from DNA. Translation is making protein from RNA on the ribosome. As we mentioned earlier, ribosome is used to make protein. Each RNA molecule is used to make one protein, and some ribosomes are bound to the membrane of the rough ER, while other ribosomes float freely in the cytoplasm. ER, we have smooth ER, which does not have ribosome attached to it. Then some part of ER are attached with the ribosome, make it rough ER. This is a Golgi apparatus, as we mentioned earlier. The function of Golgi apparatus is to finish, sort, and ship cell products. especially proteins. He works like a post office. He will label and package the protein, then secrete the protein into the vessel, then release it into the cytoplasm for utilization. The other organelle that is very important in the cell is mitochondria. Mitochondria are found in both plant and animal cells. The purpose of mitochondria is to form energy molecule ATP. Another type of the organelle that is only found in plant cell is called a chloroplast. The chloroplast is to perform photosynthesis to convert solar energy into chemical energy glucose. Both chloroplast and the mitochondria are energy production organelles. Photosynthesis is carried out in chloroplast. In photosynthesis, plant will use sunlight to create sugar molecule, and chloroplast will require supply of water and carbon dioxide to make this process. The final product, glucose, produced by photosynthesis will provide the energy to power the cell to make the plant to grow and develop. This is a structure of chloroplast. It contains cycloid, which is individual discs that make the photosynthesis occur on the surface of each disc. The fluid within the inner membrane is called the stroma. And here is a formula of photosynthesis. The cycloid within the chloroplast will use carbon dioxide and water and convert solar energy into sugar and oxygen. When you walk within the forest, you feel the air is very fresh because plant can do photosynthesis to produce oxygen. Mitochondria is located within both plant and animal cells. The purpose of mitochondria is for cellular respiration because within the mitochondria, the cells will convert oxygen and the sugar molecule into energy molecule called ATP. With that energy molecule, we can do movement, growth, and reproduction. This is a structure of mitochondria. It also has a membrane wrapped around this organelle. Inside of the mitochondria, it is a folded structure. The folding will make this organelle to be very efficient in producing energy molecule. So the chemical reaction of mitochondria is shown here. This is called the cellular respiration. It will use the substances glucose plus oxygen to make carbon dioxide, water, and ATP molecule. Another organelle within the cell is called a vacuole. Vacuole are storage sacs. Starch is stored in vacuole. Some vacuole in the animal cell is to pump water out of the cell to keep the water balanced. Some animal cells, such as sperm, has flagella. Flagella is to propel the cell through the whip-like motion to make the cell to move around. Some cell has a cilia. Cilia is a structure that can propel mucus. Cilia can be found in the cells that make the small intestine, and some cells in the trachea also contain cilia. So cilia can track the nutrients or mucus or bacteria. Plant cell has cell wall. Cell wall is for protection, maintain the cell shape and the structure stiffness. Animal cells do not have a cell wall. The cell has certain shape. This is due to the structure of cytoskeleton. Cytoskeleton is a network protein fibers that provides mechanical support, anchor, and tracks for vessel movement. Cytoskeleton network can be quickly dismantled and reassembled. That characteristic will provide the flexibility for the cell shape. Animal cell can produce extracellular matrix to help hold the neighbor cells together. The extracellular matrix are mainly proteins, so they can either anchor the cell or they can be fibers to maintain the shape of the cell. In summary, the extracellular matrix is to hold the cells together to make them into a tissue. In summary, we have talked about some organelles in the cell. We talk about nucleus, mitochondria, chloroplast, ribosome, vacuole, flagella, cilia, and extracellular matrix.